And next up, we'll have our next presentation by Emily Siniscalco from Yale University in the United States. The title of her talk today is Mucosal Immunization Induces Linked Local and Systemic Humoral Responses. Welcome,
Great,
Emily.
thank you. All right, can you see the correct screen? This should be full screen PowerPoint.
Yes, we can see it.
Amazing. All right, well, thank you for the introduction and for the opportunity to speak here today. I'm really excited to share my PhD work with you all. This is going to be sort of an abbreviated presentation, and so if you're interested in learning more about anything, please ask questions, and then there's a link to the paper at the end. Um, But in our lab, we're really interested in the gut immune system and the unique challenges that it faces, trying to balance the protection from all of these different foreign challenges, including bacteria, food, toxins, viruses, and fungi, all of which can either be sort of helpful to the host, harmful to the host, or completely neutral, depending on the quantity and context in which these challenges are perceived. And so the gut really needs to balance protection from foreign insult with homeostasis and preventing undue inflammation in the gut such that it can maintain this homeostatic balance. And one way that the gut does this, although by no means the only way, is through the production of mucosal antibodies. So, in particular, this refers to mucosal IgA, which is a dimeric antibody isotype that can get secreted across the gut barrier and bind to antigens like bacteria in the lumen of the gut. So actually, it will go from inside of your body to outside of your body and then bind to antigens that are technically on the outside of you. However, all of these antigens that are targeted by IgA, that we commonly think of as mucosal antigens, are actually also targeted by circulating IgGs, which are sort of protecting the body on a different front. And so the way that we envision this is that mucosal IgA provides local protection at barrier tissues, but if that barrier is breached, you do want to have a functioning immune system on sort of the back burner to protect you from systemic pathogen dissemination, or even if the organism is not pathogenic, just preventing it from existing in spaces where it would do harm, even if it's beneficial in the gut. So we were really curious to see how mucosal inoculation, so an exposure at a mucosal site, leads to coordinated local secreted humoral responses in the form of IgA, as well as systemic humoral responses in the form of IgG. And so, thinking about how antibodies are structured, there are two things that immunologists generally think about. We have the variable region, which determines antigen binding, and this is where the germinal center will sort of mold antibody and the antibody's ability to bind to its particular antigen through BCR mutations and increase its affinity for that antigen. And we've done some work on this, but I'm not going to be talking about the variable region very much today. Instead, I want to draw your focus to the constant region, which determines the effector function. So this determines what the antibody can do. These different constant regions engender different isotypes. So IgG and IgA are both determined by this constant region. And then this is achieved through the process of class switching. And so we're really interested in how the constant region shapes these antibody dynamics. The way that class switching works is actually quite interesting. So B cells will all start off as expressing either IgM or IgD or a combination of both of them because these are the most proximal 5' isotypes on the immunoglobulin locus. And so this is all part of the immunoglobulin locus in the genome. And then say we want to switch the B cell to IgG1. This class switching response actually occurs downstream of cytokine signals, which induce AID expression and double-stranded breaks in the switch regions of both the mu and the gamma-1 immunoglobulin genes. And then this intervening DNA is actually removed from the locus and completely excised, after which the cell will ligate the genome back together, and now it will express this IgG1. So this B cell can never go backwards because that DNA, once it's gone, is gone. However, there is the potential for it to possibly switch to these different downstream isotypes, including IgE, which has been demonstrated to be sequentially switched, and IgA, which we'll be exploring today. So we wanted to understand how this class switching response shapes these gut B cell responses. And to look at this, first we did some flow cytometry on ova-specific B cells. So these are B cells that are specific for an antigen that we gave through oral immunization. Ova is just an egg protein, short for ovalbumin. And what we notice right away is that there's a huge discrepancy between the isotypes present in the mesenteric lymph node and the Peyer's patches, which are prominent gut inductive sites. So this is where B cells will get activated and participate in germinal centers. And then this is very different from the isotypes that we see in the gut lamina propria. And this is where plasma cells will home. So this is the terminal differentiation state of B cells that are professional antibody secretors. And so we see that the cells secreting antibody are primarily of the IgA isotype in the gut, whereas the cells that are, undergoing activation in real time tend to be IgG1. And so we wanted to understand what's causing this bottleneck. Why is there a difference in isotype uh, bias across different sites in the gut? So to do this, we did a rather expensive and arduous experiment, but nonetheless very powerful, where we immunized our mice with ova and our mucosal adjuvant cholera toxin 4 times to generate this antigen-specific post-immunization response. We collected all of our tissues, including the mesenteric lymph node, Peyer's patches, lamina propria, sorted out antigen-specific, so ova-specific cells, and then sequenced them for both their transcriptomes, so we could get a sense of cell state, as well as their BCR sequencing, so we can get information like isotype, clonality, and sort of affinity maturation profiles. And all of this work was done with a ton of help from Steve Kleinstein's lab and his staff scientist, Tai Long, who have been really instrumental in the analysis portion of this. And then also my wonderful lab mates, Shuting and Xiang Yun, who did a lot of help on the bench side. So the first thing that we wanted to look at was the clonality of these B cells. Biologically, a B cell clone is the group of B cells that are all derived from the same naive precursor. So if you think about the beginning of a response, a naive B cell is going to receive a stimulus and then it's going to expand and proliferate and create all of these progeny. And what's unique about these B cells is that these progeny, unlike T cells, while they are shared by the same BDJ recombination, they also generate post recombination mutations that we can see as they diverge. So it makes this a little computationally difficult to define, but I've given the definition right here. But we are able to see that post-immunization, these clones are rather large, which is what we would expect because they've been mutating and expanding for over 4 weeks now. And we can see that several clones are over 500 cell constituents. So we can get a lot of information from these families and learn a lot about how these clones are evolving over time. But first, we wanted to perform a relatively simple analysis. We just took each clone and put it into one of 3 bins. If a clone contained at least one IgG1 cell and at least one IgA cell, we put it in the green bin. If it contained an IgA cell but not an IgG1 cell, we put it in the blue bin. And if it contained vice versa, an IgG1 cell but no IgA cells, we put it in the yellow bin. And then this is agnostic to all other isotypes. So clones with IgM can be in any of these bins, for example. And what we see is very striking. If we just look at expanded clones that have 10 or more members, which, as you can see earlier, is really most of them, we see that there's a real enrichment for clones that contain both IgA and IgG1, suggesting that these, despite being spatially distinct across our locations in the gut, are generally clonally linked through a common ancestor. And then also these clones that contain both isotypes tend to be the largest. So we see that they're much larger than clones that contain only one or the other. But just because cells are related in terms of a family doesn't necessarily tell you how they're related. So we wanted to understand how these individual cells are related within a clone. And so the metaphor I like to bring up here, this is my favorite slide, is always that of my own family. So here I've pictured my great-grandparents, my grandparents, my parents, and my siblings. And I have a different degree of a relationship with each of these people, from my grandparents, great-grandparents who I've never met, but I've heard stories about and who I share some of my genetic information with, to my grandparents who I'm close with but have very different life experiences, and then my parents and siblings, and so forth. And so we wanted to see similarly, are the IgA and IgG1 B cells within a clone very unrelated, but still linked, sort of like they would be grandparents or great-grandparents or just distant cousins of each other? Or are they more related like siblings and share a lot of the same experiences, and we can infer more from those relationships? And so to analyze this, we generated essentially phylogenetic family trees for each of our B cell clones. And I know that there's a lot of information here, but what I want you to pay attention to is, first of all, the color. So the color here denotes isotype, and the green is going to be IgG1, and the blue is going to be IgA. The other thing that's important to note is that these cells are, their relationships are determined based on their BCR mutations. So the closer two cells are on the tree, the more common mutations they have between them. And as you go from left to right, the cells get more and more mutated. So this black box is the germline ancestor that should have been the BCR of the naive B cell. And then as you go further to the right, these cells get more mutated. And so one thing we can appreciate right away is that the blue and the green cells are very intermixed within the tree. It's not such that there is a green branch that is all IgG1 and then a blue branch that is all IgA. Instead, these cells seem to be very sort of intermingling within the lineage of the clone. We also notice a few common patterns. So first, which we very creatively termed pattern one, is the existence of these IgG1 cells that have the same BCR mutation pattern as IgA. So these have identical BCR heavy chains. And what's striking about that is that it means that all of the mutations that they've acquired up until this point were highly unlikely to have occurred separately, but were much more likely to have occurred in a common ancestor of these two cells. And if we carry this pattern out, we think that it's unlikely, since most of these IgA cells are plasma cells, they're unlikely to acquire any more mutations. So they're going to stay sort of static at the same horizontal point on the tree, whereas these IgG1B cells are going to continue mutating and continue moving towards the right. And so we've termed that pattern two. And if we just look at this upper left here, we can see that expanding this box reveals essentially that same pattern, showing us that we're observing similar things that support our hypothesis in the same tree. And we don't just see this once, but we see it several times, both in this tree and in one of the almost 60 trees, or many of the almost 60 trees that we've generated from this data set. So what we're proposing here is not necessarily direct class switching like I demonstrated earlier, but rather something called sequential class switching, where a cell will class switch from IgM to an intermediate isotype. And then we're saying here that this intermediate isotype is likely IgG1. This IgG1 cell will undergo the germinal center maturation process, explaining why there's so much of it in these gut inductive sites. And then some of the progeny of this IgG1B cell will go on to class switch to IgA. So this allows for both that linkage of IgA and IgG1 that I talked about at the beginning, as well as sort of honing the BCR repertoire prior to switching out these constant regions for modular changes. But we also wanted to be able to prove this in a setting that was not just in our single cell data set. We wanted to take this into the mouse and see if this was actually happening. So one way that we can do this is by taking IgA plasma cells from the gut and actually looking at their immunoglobulin locus to see if there are remnants of IgG1 expression in those B cells. And what we see when that occurs is that there's a sequence that aligns with the S gamma 1, the switch region of the IgG1 locus, sandwiched in between the S mu and the S alpha of this constant region. And when we PCR amplify the immunoglobulin regions of these IgA plasma cells in the gut, we do indeed find amplicons that have this IgG1 remnant, suggesting that sequential class switching is something that's happening in the gut, particularly after mucosal immunization. Now, I like to leave every talk thinking about sort of my gut feelings, pardon the pun, why make this IgA from the IgG1? And I sort of spoiled the answer way back at the beginning of the talk, but we think it's really important for linking this mucosal and systemic protection, given that in mammals, the mutation and the affinity maturation of the BCR is so important to hone the response, it makes sense to only want to do it once. And so by mutating and affinity maturing the IgG1 B cell in the germinal center, you then free that response to be able to modularly affect the effector function of those antibodies just by swapping out the constant region after you've already set the variable region. So this allows for antibodies that have the same antigen binding to antigen to both provide mucosal protection and systemic protection at different sites. Now with that, I'd like to thank all of the different people that made this work possible. My mentors, Steph Eisenbarth and Joe Kraft at Northwestern and Yale, respectively, have been incredibly helpful throughout the many, many years of this project. Everybody whose name is in bold here contributed data. Everybody whose name is not in bold contributed irreplaceable moral support and help. A lot of the people on here, I didn't get time to share their data, including those from the Westman lab and those from the Crotty lab, which provided human data supporting our hypotheses. And then, of course, I'd like to thank all of you for listening. I am happy to take any questions. And if you're interested in reading the paper, it just came out in Immunity, and you can scan the QR code here.
Thank you so much for your excellent talk, Emily. One of the questions from our audience is that what are some potential next steps for your project?
Yeah, so I think that probably the biggest next step, unfortunately, I'm leaving my PhD lab very soon, so I won't get to do all of these things. But should I be, writing a grant or trying to think of things to do next, I would love to know if sequential class switching is happening in other contexts. So for instance, we know that IgG1 can also class switch to IgE in the context of food allergy. And this is important for generating high affinity IgE antibodies that are pathogenic in that situation. We've now demonstrated IgG1 to IgA class switching, but I think that there's nothing stopping class switching to other isotypes, even other IgGs. And so I think it would be really interesting to sort of explore that a little more. We're also curious about the implications of how an IgG intermediate changes the IgA antibody response, but that's been a little difficult for us to pursue so far.
Definitely. And I guess since you mentioned it, another question I had was like, what are some signals in the gut microenvironment that could potentially drive the sequential class switching from IgG1 to IgA?
Right. So we think that the sequential class switching is driven in the exact same way as a direct class switch event would, except for the fact that it's preceded by a class switch to IgG1. So to get to the IgG1, you need IL-4 signaling that will open up that IgG locus, and then you can get the AID expression and the class switching there. We think that that must then be spatially restricted from the IgA class switching signals, and there should be something that's sort of directing the cells to class switch to one or the other. IgA class switching in the gut is very interesting. It occurs pretty specifically in the Peyer's patch subepithelial dome, so a really unique, like sub microscopic region that is very DC rich, and it contains a rather complicated pathway where TGF beta secretion by B cells is activated by DCs, which then signals back to the B cells and allows them to class switch to IgA. And so we've demonstrated in our work that there are post-germinal center IgG1 B cells that do migrate to the subepithelial dome. So we think that sort of restricting the IgA class switching to that region allows for B cells in the follicle to mainly be directed towards the IgG1 isotype, enter the germinal center, and then go to the subepithelial dome and switch to IgA.
Interesting. Another question from Dr. Paula Licona Limon is that did you check if there was any compartmentalization in this differential isotope used within different regions in the gut? Is this shaped
Mm.
by microbiota in any way?
Yeah, so I'll answer the microbiome question first because that one is simpler. We have not delved into the microbiome at all. What I can say is that we see sort of links between IgA and IgG1 both in the mice, and we see that in human bone marrow and human nasal lymphoid tissue. So we think that this is sort of a broader evolutionarily conserved response as opposed to something that's being specifically driven by the microbiome. We intentionally kind of haven't waded into those waters too much, but it's definitely an interesting question. In terms of the compartmentalization, the gut is very interesting in terms of its isotype compartmentalization. If you're just looking at total B cells, the IgG1 is much more enriched in the mesenteric lymph node than it is in the Peyer's patch. However, we were really surprised to see in the flow data that I sent you or that I showed you guys earlier that the Peyer's patch has a lot of IgG1 when it comes to these antigen-specific B cells. So something that's important to consider with the gut is that most of the antibody responses that are happening at steady state in response to the microbiome and in response to the foreign antigens we consume, like our food. And the stuff that we're observing post-immunization is a relative minority of that. So we're essentially pulling the needle out of the haystack and finding that the needle is having different properties to the haystack at large. And so we're not sure exactly how prevalent sequential class switching is to the like antimicrobial response and to the homeostatic IgA response, but this is very specific seemingly to the post-immunization response in the context of inflammation.
Thank you so much, Emily. I didn't have time to go through all the questions in the chat, but if you want, you can feel free to answer them in the chat.
Yeah, I'm happy
Well,
to.
yeah, thank you so much.